Okay, awesome. So we'll keep this informal. I hear that lots of your talks are on birds, so we'll have a winged animal talk today, but not a bird, a bee talk. So as Kayla mentioned, I am a veterinarian and I also study honeybees, and that might seem like an unusual combination, but hopefully by the end of this talk, you'll understand why those two fields fit together quite nicely. So we're going to talk about sustainable agriculture, and I thought as an example, I'd start from my own life. So this is my dad, and he is cutting hay on our farm, which is just north of Yorkton, Saskatchewan. And the hay crop that he is cutting here is a special one. It is a pollinator-friendly hay crop that was planted with support from Ducks Unlimited, who has this marginal acres program that encourages farmers to seed marginal land to pollinator-friendly forages. And so this can benefit pollinators and also provide forage for livestock. And you can see that my dad's got a pretty old swather and tractor there, but it seemed to do the job. And this hay crop, when it was in flower, provided nutritious nectar and pollen for the 3 honeybee colonies that we have on our farm. And as well, it provided some nutritious forage for our small cow herd. So I would tend to agree that this is maybe an example of sustainable agriculture. It's supporting multiple species, cattle, pollinators, and indirectly humans. But I'll leave you to consider if this is a sustainable system and whether it fits this definition of sustainable agriculture. So according to the United Nations Food and Agriculture Organization, to be sustainable, agriculture must meet the needs of present and future generations while ensuring profitability, environmental health, and social and economic equity. And so for me, the important part about this definition is that agriculture is meeting the needs of present and future generations. And so the example of my family farm, that would feed a small number of people through those agricultural practices. But if we think about the human population as a whole, by 2050, our planet is expected to have around 9 billion humans. And I'm not sure that the sustainable farming model that my family is practicing would necessarily feed all of these people. And so in order to meet the needs of future generations, most likely we are going to have to produce more food with less resources. And so one way that we can do that is through agricultural intensification. So this is a blueberry field in British Columbia, and you can see that this is a monoculture. It is the same species of blueberry for many acres. And in order to keep this blueberry field healthy, frequently farmers need to treat with fungicides. So they're spraying this blueberry crop in order to protect the health and yields of this blueberry crop. And as well, in order to pollinate the blueberry crop, they need migratory beekeepers who bring in colonies each spring, and honeybees contribute about 90% of the value of our blueberry crop in Canada every year. So they are essential for production of this fruit. But perhaps you can see some potential problems with this agricultural intensification. One of the positives of agriculture intensification is that it's going to increase yields and feed more people. But at the same time, it's going to require increased pesticide use, and possibly it's going to result in increased pollinator mortality. But that remains a question that my lab group is investigating. So this is our team at the vet school who is investigating questions surrounding pollinator health and how that relates to the agriculture systems that pollinators are supporting. And so with help from our funding partners, we are trying to investigate whether we can improve pollinator health and in so doing enhance the sustainability of agriculture in Canada. So I'll just give a brief outline. I'm going to start by talking about the role of pollinators in sustainable agriculture, and then I'll speak specifically about the role of pollinators in our canola industry, in our blueberry industry, and then in our commercial beekeeping industry. So to start with, we'll focus specifically on honeybees. And we know that honeybees are important pollinators. They are pollinating about 2/3 of the world's leading food crops. And they're contributing on average about $5.5 billion worth of pollination services to Canadian agriculture every year. And so this includes 90% of the value of our blueberry and apple crop, as well as contributing over $3 billion to canola through production primarily of hybrid canola seed, which is more productive, as well as through increased yield and quality of commodity canola. But that's just honeybees. We need to remember that we also have 387 species of wild bees here on the prairies, and the value of their pollination services to Canadian agriculture is only just beginning to be understood. But in many ways, honeybees can be a very good model for wild bee species. And so the focus of my lab is primarily on honeybees. And honeybees have been facing some challenges in recent years. And so for the last 15 years in Canada, beekeepers have been experiencing unsustainable overwinter losses of their colonies. They're losing between 15 to 35% of their colonies each winter. But last winter was a real exception. We had the highest overwinter mortality in Canada ever recorded, and Canadian beekeepers lost over 45% of their colonies last winter. And so you can imagine, to try and stay in business as a beekeeper when you're losing nearly 50% of your animals every year can be very challenging. And so what are the causes of this overwinter colony loss? Well, we can group them into the typical biotic and abiotic stressors you might think about in other systems. And so for biotic stressors, certainly there are a number of honeybee diseases that include nosema. This is a gut parasite of honeybees, American foulbrood, bacterial disease of honeybees, and varroa mite, probably the leading pest of honeybees that's not only weakening colonies, but transmitting viruses within the colony. And then we also have host factors like reduced queen quality or weak colony strength and fall. And we have environmental factors. So these can include weather, nutrition, so lack of floral diversity in the landscape, as well as pesticide exposure in the environment. And so I'm going to use pesticides as an example of how honeybees interact with our agricultural systems and how we might optimize our pesticide use to protect colony health. And so specifically, we'll now talk about pollinator health as it relates to the canola industry in Canada. And so canola is now our top crop in Saskatchewan, And greater than 95% of the canola grown in Saskatchewan is grown from seed that is treated with a neonicotinoid insecticide. And so the most commonly used neonicotinoids on canola are thiamethoxam and clothianidin. And then imidacloprid is probably the third most common neonicotinoid in Canada, but not used on canola. And so these are systemic insecticides. They're taken up into the growing tissues of the plant where they're present at low concentrations in the pollen and the nectar and present a route of exposure for honeybees. And so we know honeybees are foraging on canola. It's a very bee attractive crop. At the same time, we know that they are being chronically exposed to low concentrations of neonicotinoids when they forage on this crop. And so what is the effect of this chronic neonicotinoid exposure on our honeybees? Well, one thing we found is that chronic low-dose exposure decreases honey production. And so the blue bars here are representing colonies that were exposed to 20 nanograms per gram of neonicotinoid chronically over 9 to 12 weeks. And this length of exposure is realistic if we would consider subsequent canola blooms and blooms of other perhaps non-target species that are contaminated with neonicotinoids. And what we can see is that the blue bars represent a significant decrease in weight gain of those chronically exposed colonies, and weight gain is just a proxy for honey production. So chronic neonicotinoid exposure at high environmental concentrations of 20 nanograms per gram will significantly reduce the honey production of those colonies. As well, chronic neonicotinoid exposure can increase overwinter colony loss. And so here we're looking at percent winter mortality of colonies exposed to 20 nanograms per gram. Sorry, 20 nanograms per gram of thiamethoxam and 100 nanograms per gram thiamethoxam. And so at 100 nanograms per gram, we see a 55% increase in overwinter loss. 100 nanograms per gram, though, would be a positive control that would not be environmentally realistic. And then at 20 nanograms per gram, we see a 15% increase in overwinter mortality. So this 15% increase is not statistically significant. But if you're a beekeeper, I think a 15% increase in your overwinter loss would be significant to your bottom line. So it does perhaps raise concern. So 20 nanograms per gram, that seems to be sort of the upper threshold where we're going to see colony level effects of this pesticide exposure. So is that 20 nanograms per gram, is that realistic in terms of what we see in the environment in Saskatchewan? So there's been some work to look at what are the levels of neonicotinoid contamination in our honey. And sort of the global worldwide average is about 0.3 nanograms per gram of the neonic thiamethoxam. And there's been one study looking at the concentrations of neonics in Saskatchewan honey. And this study was done here at the U of S, and they found 17 nanograms per gram. So that is 57 times the global average in honey collected from the Saskatoon area. But this was a small study, only around 30 samples of honey were analyzed. And so I'm not sure that that's representative of the contamination that we see in the province as a whole. And so, so we need to investigate that further. And the other interesting thing is that despite these apparently high levels of pesticide contamination in Saskatchewan honeybee colonies, we have the most productive honeybee colonies in Canada. And so on average, Saskatchewan honeybee colonies are producing over 200 pounds of honey per colony. So we really are the leaders. And most of that honey is canola honey. So we know they're being exposed to neonicotinoids, and yet they're extremely productive. So there seems to be perhaps a little bit of a discrepancy there. And so to investigate that question further, we want to look at what the actual levels of pesticide exposure are in honeybee colonies across Saskatchewan. And so we have a postdoc, Marcelo Camilli, who is going to be doing this study this year. And he's selected 15 canola fields across Saskatchewan. And as controls, he's selected 4 sites in the boreal forest. And he did a pilot study last summer shown by the red bees here. And what he's going to be looking at, he's going to be putting 4 honeybee colonies at each of these sites. And he's going to be sampling bees, honey, pollen, and soil from all of these sites and analyzing those samples for 120 different pesticide residues to get a really comprehensive look at what the pesticide exposure is of these colonies. And then at the same time, correlate that with the weight gain and productivity of these colonies. As well, we don't want to ignore the fact that we have wild pollinators in Saskatchewan as well. And so as part of that, we're going to be doing some surveillance for wild pollinators at each of these sites and seeing if pesticide exposure may have an impact on abundance and diversity of wild pollinators in agricultural versus non-agricultural landscapes in Saskatchewan. So that's sort of some of our work in the canola industry. And now I'll transition to talking a little bit about the blueberry industry and our work on both the West Coast with high bush blueberry production and the East Coast with low bush blueberry production. So recently there's been a lot of media coverage about whether or not blueberry pollination poses a health risk to honeybees. And so we wanted to gather some more evidence to understand this relationship. And at this time, blueberry growers are having a hard time attracting beekeepers to pollinate their crops because of this perception that honeybee health declines when they go to pollinate blueberries. And so part of the reason that beekeepers are reluctant to go to blueberries is because they're losing a lot of their colonies over the winter, and blueberry pollination typically occurs on the West Coast in the early spring. And so oftentimes beekeepers just don't have enough colonies to send in the early spring. And it's logistically difficult. If the colonies are kept in Alberta or Saskatchewan, they need to load them on a truck and transport them into blueberries in BC, which you can imagine can be very stressful on the colony. And as well, beekeepers will sign up for pollination contracts where they agree to bring a certain number of colonies. And if due to their overwinter losses, they don't have enough colonies, then there's pressure for them to split those colonies, divide them into 2, which will allow them to meet their contract requirements. But the downside is that those colonies will be weaker and perhaps more vulnerable to disease. And one of the diseases that we see most commonly in blueberry pollination is European foulbrood disease. And so EFB, or European foulbrood, it's a bacterial disease of honeybee larvae. And the most common clinical sign is an abnormal brood pattern. And so they refer to this as a shotgun brood pattern because it looks like someone took a shotgun and destroyed the larva, if you compare to this nice uniform brood pattern here. So you can imagine having death of the larva means fewer bees available to pollinate the crop, and the colony can, dwindle and die as a result of this. And so, as I mentioned, it's a bacterial disease. The bacterium is known as Melissococcus plutonius. And we have a model in my lab to study this disease. And so we are able to infect larvae in the lab with 50 colony forming units of this bacteria. And then we can chronically expose these larvae to, for example, fungicides used in blueberry production, and see how fungicide exposure might impact the larva's survival from the disease. To understand whether or not, for example, fungicide exposure in blueberry pollination could be a predisposing factor for poor colony health. And so this is an example of some of our data. So on the y-axis, you have percent larval survival, and then on the x-axis, you have time in days. And so as our control group, we have in blue larvae that were fed formulated fungicide diet. So it contained 4 different fungicides used in commercial blueberry production at environmental concentrations. And you can see that those fungicide exposed larvae had excellent survival, survival that was comparable to the larva fed non-contaminated control diet. And then we can go ahead and infect those larvae with the bacteria. And as expected, we see a decrease in survival in those infected control larvae. But what's interesting is that if we infect the larvae and then expose them to 4 different fungicides, we see a significant decrease in their survival relative to the infected control group. And so this would suggest that perhaps chronic fungicide exposure may decrease survival from European foulbrood and could be a predisposing factor for this disease during blueberry pollination. But it's interesting that we don't see this effect of fungicide exposure when we use the fungicides individually or in groups of 2 or 3. It's only when larvae were exposed to 4 fungicides in combination that we saw a significant decrease in their survival from the bacterial disease. So data like this can perhaps encourage conversations among blueberry growers and beekeepers to potentially avoid those combinations of fungicides that may have deleterious effects. Because at the end of the day, we understand that blueberry growers need to use fungicides to protect their crops, but they also need pollinators. And so perhaps we can find some kind of a compromise where we can use fungicides, but use them in a way that protects pollinator health. And so the last thing I'll touch on is pollinator health and the beekeeping industry itself. And so in this video here, I'm wondering, can anyone tell me what we're doing to this colony? Maybe Loki, do you know? Yeah, so that was an antibiotic treatment of a colony in our lab. And this is quite a common, widespread practice in North America. And what's interesting is that in the European Union, it is illegal to use antibiotics in beekeeping. And here in North America, we're using a lot of them. So if you are to treat a colony with oxytetracycline, which is the most commonly used antibiotic in North America for beekeeping, if you follow the label, you will be treating it with 1,200 milligrams of active ingredient. And that would consist of a spring treatment and a fall treatment outside of honey production so that you can avoid residues in honey. And so if we think about the total number of hives in North America, it's just over 3 million. And on average, we know that about 66% of those colonies, or about just over 2 million colonies, are treated with antibiotics every year. Then doing the math, that means that we're using around 2,500 kilograms of active ingredient every year. And the most commonly used formulation is this oxytetracycline A product. So that would mean that we're using over 46 tons of oxytetracycline A in colonies in North America every year. And in Europe, they are using none. So perhaps we need to reevaluate our use of antibiotics in beekeeping and make this perhaps a little more sustainable. Since 2018 in Canada, beekeepers now require a veterinary prescription in order to access antibiotics for treatment of their hives. But in order for this to be a positive change, we need some more evidence to understand when and why and how to use antibiotics in beekeeping so that we can preserve them as an effective tool when we need them, but also so that we can minimize the incidence of antimicrobial resistance. Because perhaps not surprisingly, we are starting to see antibiotic resistant strains of bacteria appear in our honeybee colonies. So I'll remind you again about European foulbrood caused by Melissococcus plutonius. And when we talk about bacterial antibiotic resistance, oftentimes we use the term minimum inhibitory concentration, or MIC. And so the MIC is essentially the lowest concentration of antibiotic that you need to inhibit the growth of the bacteria. And so to consider Melissococcus plutonius sensitive to oxytetracycline or susceptible, you should have an MIC of 2.5 micrograms per mil of oxytetracycline. So this 2.5 micrograms per mil should inhibit the growth of M. plutonius to consider it sensitive to oxytetracycline. And so what are the MIC values for M. plutonius around the world? Well, 20 years ago, they looked at M. plutonius in Australia and found that the MIC was between 1 and 2. Great, it seems to be susceptible to oxytetracycline. They also looked in the UK and the MIC was around 4, so a little bit above that susceptibility breakpoint, but still not very high, and probably within the hive, this antibiotic is still working. Let's fast forward now to 2019, where our lab looked at the sensitivity of M. plutonius in a colony from British Columbia that was involved in blueberry pollination. And we found that the MIC was 32. So significantly higher than the susceptibility breakpoint. And this would be an example of a resistant isolate of Melissococcus plutonius. So based on our laboratory testing, this would suggest that we have antibiotic resistance, and we're now finding that it's actually quite common. So on this Petri dish here, we have 25 different isolates of Melissococcus plutonius from around Western Canada. And this Petri dish contains oxytetracycline at the breakpoint concentration of 2.5 micrograms per mil. And you can see that we have bacterial growth in every single quadrant. So antibiotic resistance appears to be widespread. And so we need to perhaps reevaluate how we are using antibiotics in the colony and perhaps consider whether or not we need to be using other antibiotics in place of oxytetracycline, because currently for European foulbrood, oxytetracycline is the only approved antibiotic. There are no other options currently. And then I'll just now touch on another bacterial disease of honeybees, American foulbrood disease. And so this disease has similar clinical signs to European foulbrood in that it causes this shotgun brood pattern. But the difference with American foulbrood, which is caused by a bacterium known as Paenibacillus larva, is that it's going to affect the pupa more than the larval stage. And this dead pupa is going to form a scale that you can see shown here by the arrows. And this scale contains billions of spores and billions of extremely resilient spores. And the only way to get rid of this disease is essentially to burn the colony. And so because these spores are oftentimes present in colonies at a low level that does not cause clinical disease, beekeepers treat prophylactically with antibiotics in spring and fall to prevent outbreaks of the disease. And so once again, you can imagine that regular treatment with oxytetracycline is going to breed oxytetracycline-resistant Paenibacillus larvae. And that's exactly what we found. And so this graph here is showing you the minimum inhibitory concentration of oxytetracycline for over 700 different isolates of Paenibacillus larvae that we collected from 52 different beekeepers in Saskatchewan. And you can see number of isolates on the y-axis here. And so the good news is that about 90% of them are sensitive to oxytetracycline. And so beekeepers could continue using this antibiotic and it would be effective to prevent American foulbrood in their colonies. We have about 1% of isolates that have intermediate sensitivity to this antibiotic. But concerningly, we have 9% of isolates which are clinically resistant to oxytetracycline. And so these beekeepers are using other antibiotics. In most cases, they're using Tilazin to control this disease, which still remains effective, but Tilazin would be considered a more medically important drug for humans. And so we'd want to minimize our use of this drug in agriculture to protect ourselves. And furthermore, Tilazin treatment is associated with increased risk for residues in honey. So it is not the ideal antibiotic, but these beekeepers, because of their resistant isolates, have no other option. And also concerningly, it appears that antibiotic-resistant Paenibacillus larvae is spreading in Saskatchewan. And so this is a modified map of Saskatchewan to protect beekeeper confidentiality. But each of the pie graphs here represents a different commercial beekeeper in Saskatchewan. And the red indicates oxytetracycline resistant Paenibacillus larvae. The yellow is the intermediate resistant. The green are the oxytetracycline susceptible. And so you can see that in the Northeast, we have the highest concentration of antimicrobial resistant strains of P. larvae, but it seems to be spreading to the Northwest. And we've done whole genome sequencing on these isolates, and they are extremely similar and carrying the same tetracycline resistance genes. So it would suggest high relatedness and most likely spread of the same resistant strains across Saskatchewan. The concern, though, is that we don't currently have resistant isolates detected in the Saskatoon area or southern Saskatchewan. And you can see that in the Saskatoon area, we have a lot of beekeepers. And so we're concerned that these resistant strains may continue to spread. And part of what our lab is doing is ongoing surveillance to monitor and hopefully alleviate that spread. And so one of the ways that we can potentially reduce the spread of antimicrobial resistance is to reduce our use of oxytetracycline to reduce the selective pressure for resistant strains. And we can reduce our use by potentially using more evidence-based antibiotic therapy. And so one way to do that is to look at the number of American foulbrood spores in honey and allow the spore concentration in honey to guide your treatment decisions. And so this graph here shows American foulbrood spores per gram of honey. And we had, again, 52 different beekeepers participate in this study in 2019 and 2020. And so we literally just cultured their honey samples to determine their spore level. And we grouped them into risk categories. So we considered those beekeepers that had 2 spores per gram of honey or less to be low risk for an outbreak of American foulbrood, and those between 2 and 100 spores per gram of honey to be at sort of intermediate risk, and that those at more than 100 spores per gram of honey to be high risk for American foulbrood. And then we looked forward into the future, and based on those risk categories, looked to see how many beekeepers had outbreaks. And so what we found, the red dots here are representing outbreaks of American foulbrood. We didn't see any new outbreaks of AFB in our low risk category. And we saw the highest incidence of AFB in our high risk, and only some AFB in the intermediate category. So this would suggest that our test is a good one, and that perhaps beekeepers can now use this test to inform their antibiotic use. So if you're in the low risk category, perhaps you don't need to be treating with antibiotics to prevent this disease. If you're in the intermediate category, maybe you just need to do some really good monitoring and maybe some other disease management practices in combination with antibiotic therapy as required. And then if you're in the high risk category, you probably should be treating with antibiotics. And so we think that maybe this kind of testing could reduce our reliance on antibiotics in the beekeeping industry and correspondingly reduce the selective pressure for resistant strains. And at the end of the day, we have our own self-interest in mind when we talk about antibiotic resistance. Currently, I believe the estimate is that globally we have about 5 million deaths per year from antibiotic-resistant infections in the human population. And so one way to preserve the efficacy of antibiotics for ourselves is to reduce our reliance on them in agriculture. And so that's what our research group is attempting to do. So to summarize, pollinators are essential to sustainable agriculture in Canada, and our research focuses on optimizing intensive agriculture practices to promote pollinator health. And specifically, we're interested in looking at modifying pesticide use and antibiotic use to create a more sustainable agriculture industry that protects not only crop health, but also pollinator health and human health ultimately. And I think when we think about sustainable agriculture, it really is important to remember that we're thinking about the future, that future generations and young people like yourselves are going to be sort of the beneficiaries of our food systems and how we organize them. And so we're very much interested in training new scientists and new beekeepers to understand the challenges that we face in beekeeping and agriculture generally. And hopefully that can be part of a more sustainable future. So this is our honeybee disease elective course for veterinary students. And so every spring we run this course and teach new veterinarians about honeybee diseases and how to manage them. And certainly we are always recruiting new graduate students for our lab. So, I have some shameless promotional recruitment material here. And if you're interested in potentially studying pollinator health, we take all types of people, not just veterinarians. And even if you want to come help us in the field, we had Loki out there helping us wrap our colonies for winter this fall. Certainly just contact me and I'd be happy to bring you to the field. So with that, I'm happy to take any questions.
Thank you so much, Sarah. That was a really great presentation. So if anyone in the room has questions, I will come around and pass the mic to you. And if anyone online has questions, you can feel free to unmute yourself. But if you're a little shy, you can also throw it in the chat and we will read them out and Sarah can answer them for you. So, any questions?
Awesome presentation. I have two questions. The first being, I think you touched on this just briefly, but is there studies on these antibiotics being found in honey samples in any concentration or in even concerning concentrations?
Yeah, so honey in Canada is regularly tested for antibiotic residues, particularly because we're an exporting country. So other countries want to know what our residues are. And so there are maximum residue limits set for honey, and our honey cannot exceed those. And so beekeepers are very good about avoiding antibiotic treatment during the honey flow. And as well, when we treat a colony with antibiotics, we're only treating the brood chamber, or the part of the colony that isn't being harvested for honey. And so we'd never treat a colony that has so-called honey supers, or boxes that have honey in them for human consumption. So yes, it's very tightly regulated, but that's not to say that there isn't concern. So as I mentioned, ptylosin has a longer residue period. So we currently only recommend ptylosin treatment in the fall, because treating in the spring prior to the honey production season can present a risk. And did you have a second question?
Got it.
Okay.
It was about just why you only treated the one little section, but it's the brood chamber.
That's right. Yeah. And you're treating sort of at the back of the colony, because at high concentrations, the antibiotic can actually be toxic to the brood. So you sort of want to avoid the brood nest. And then also the antibiotic is light sensitive. And so you want to protect it from light, and you can feed the colony sugar syrup with antibiotics, or the example that I showed there was just mixing it with powdered icing sugar, which would be the most common way we would treat.
It looks like we have one question in the chat. So Christy has asked, in your current provincial pollinator sampling, what pesticides are you looking for?
Yeah, so we're just doing the most comprehensive pesticide screen that we can. So I think currently our plan is to go with the University of Guelph, which has between 120 and 250 different residues that we can look for. So they would range from neonicotinoids and various fungicides and their metabolites.
Yeah, cool talk. I'm curious, is the reduction in honey produced by the honeybees a result of just the honeybee mortality, or is the neonicotinoids actually affecting the pollinating behavior of the bees that are surviving? And I have a follow-up question after that.
Yeah, and so probably both. So what I didn't show you is that those colonies that produce less honey also had fewer bees. So probably there was an effect on population size, but there's many laboratory studies that show effects on behavior from sublethal exposure to neonicotinoids. So it could affect their ability to, for example, return to the colony from a foraging trip. The interesting thing about studying honeybees is that they're a superorganism, so they can compensate, they can be very resilient. And so I think that's why it's important to have a combination of field and laboratory studies, because an effect that you see in the lab may not always scale up to an effect at the colony level because of this intrinsic resilience of the superorganism.
And the second question to that is, you guys detected neonicotinoid in the honey. Is there evidence of it elsewhere in the colony, like in the wax that the bees are using, or the royal jelly that gets fed to future queens that might affect queen health going forward?
Yeah, good question. So you're right, it can be in all hive matrices, the pollen or bee bread, the wax, the royal jelly. Typically, like royal jelly, the concentrations are extremely low because that royal jelly is processed through the hypopharyngeal glands of the nurse bees. And so taking, say, a contaminated pollen via a nurse bee into the royal jelly, the translocation of neonicotinoids is going to be at extremely low level. But yeah, all hive matrices present a potential exposure risk. But as we know, the dose makes the poison.
Cool, thanks.
Oh yeah, go for it. If you have your hand raised, you can go ahead and speak.
Oh, thank you. Can you hear me?
Yes, we can.
Okay, super. Well, thanks, Sarah. That was a great talk. you talked about this interactive effect between fungicide and disease on the overwinter survival. I'm just curious about, do you know how weather interacts with that interaction and what that might mean in the broader context of climate change?
Good question, Elsa. Yeah. So you're right that weather can certainly impact susceptibility to disease, and we didn't control for that necessarily. And yeah, it's just another sort of abiotic factor that can influence how a pesticide might, say, immunosuppress a colony and cause them to have reduced overwinter survival. So what I expect, with a unpredictable climate, that we might have increased overwinter losses and increased interactions between bees and pesticides with negative outcomes. Sure. Certainly, again, this idea of like a superorganism during the winter, that honeybee colony is forming a cluster. They're not hibernating, but they're thermoregulating in that cluster. And so it can be -30 outside and it can be +30 in the center of that cluster. So incredible ability to thermoregulate, but are they going to be able to do that in the face of multiple stressors such as neonicotinoids, disease, and unpredictable climate? I don't know. I hope so, but I think any way that we can alleviate some of those stressors is going to make them more resilient.
I was curious, as far as the testing for the bacterial, towards the end, you talked about the testing for the spores. Is that something that is like relatively easy to do for a beekeeper, or do you expect any sort of, I guess, barriers to making that a common practice?
Yeah, good question. So our beekeepers in Saskatchewan are terrific. The only reason we were able to do that study is because we had great buy-in and they were all willing to send in their honey samples. So there's no problem there. The problem, though, I foresee is the time that it takes. So it takes about 7 days to culture the honey sample, and that may not coincide with a timeline for a beekeeper to make a treatment decision, right? They might need to make a treatment decision right away, and you're going to tell them, well, it's going to take a week for me to culture your honey, determine your spore level, and whether you have resistance. And so it would be great if we could make a COVID test, right? That a hive side test that could tell you the number of spores you have in your honey. Currently, there is a COVID test that can tell you whether or not you have American foulbrood, but it can't tell you your spore level. And so our lab is currently working on developing a PCR approach to determine the level of spores in honey, because that would be a lot quicker than doing bacterial culture. The problem that we're currently experiencing is that, number one, honey is sort of a difficult medium to work with and to extract the spores from. And sometimes when we're talking about 2 spores per gram, that is a very low detection threshold. And the CT values, or for quantitative PCR, are often very high with that low spore level. So the sensitivity becomes an issue. So yeah, we're working on it, trying to improve the diagnostic efficiency of the tests so that it can be a realistic option for beekeepers when they're making decisions.
Thank you.
Anyone else? I also have a question. So you mentioned in Europe that antibiotics are banned for beekeeping. So how are they managing diseases like the European foulbrood there without those antibiotics?
So great question. So one big difference between Europe and North America is that hobbyist beekeepers in Europe are much more common. So it would be much more common to have 10 or 20 sort of backyard colonies, versus in North America, we're talking about commercial beekeepers with thousands or tens of thousands of colonies. So when you're dealing with sort of a hobbyist situation, it's much easier to do individual hive medicine, where you can go and inspect every single colony. And if you think it's sick, you can do some just beekeeping husbandry practices that don't involve antibiotics. So for example, with the European foulbrood example, it's often a disease of stress. So if you were to give that colony some supplemental nutrition, or if you were to give that colony some frames of brood so they had more adult bees to nurse the sick larva, or you could do what's called a shook swarm, where you literally just shake the bees into new equipment so that and burn that diseased equipment. All of those practices are very effective ways to address that bacterial disease without using antibiotics. The problem is that those kind of practices are very labor intensive. And when you have tens of thousands of colonies, it does not sometimes become realistic, and hence the antibiotic use. So is there another way? Yes, there is. But we get into that problem where we've got mouths to feed, we need intensive agriculture, and what are the solutions? Can we make a compromise?
Thank you. Anyone else have any questions, either in the room or online? Seeing none, I think we will wrap up. Thank you so much, Sarah, for coming and giving this talk. I really enjoyed it, and I'm sure everyone else did too. Our next seminar will be on March 3rd, and that's Iga Stasiak with Managing Wildlife Health, a Provincial Agency Perspective. So come join us on March 3rd for our next seminar. Thanks so much, everyone.